Viruses are tiny, usually even tinier than bacteria. So how in the world do we study something we can't see? My name is Hannah Gavin. I'm a microbiologist at MIT and Harvard who studies bacteria infecting viruses. These viruses have their own name, bacteriophage, meaning eaters of bacteria. As far as we know, bacteriophages can be found anywhere bacteria live, from your kitchen counter to the boiling geysers of Yellowstone. The phages I'm interested in live in the ocean. Viruses are the single most abundant biological entity in the ocean. Even though scientists have studied phages for more than a century, there are still so many outstanding questions about the basic biology and ecology of these viruses. How do phages attach to and kill their host bacteria? How do phages influence bacterial populations in the ocean? How do bacteria resist phage infection? Scientists study viruses in the lab in many different ways, but I'm going to show you how I do it. Since phages are so tiny, you might imagine that I use a microscope for all my experiments. But actually, I hardly ever use microscopes in my work. That's because I can use indirect methods, that is, the activity of the phages, to detect them instead. This petri dish is covered in so many bacteria that they form a lawn. That's the opaque covering over the whole plate. The clear spots contain aggregations of thousands of virions, or individual viruses, that have killed some of those bacteria. This is how we can see phages without a microscope. Now I'll show you how I would use this technique to learn something new. The first thing I have to do for any experiment is grow bacteria for the viruses to eat. I take a small amount of frozen bacteria and spread them onto a nutrient-containing jelly called agar. Initially, there are too few bacteria to see, but I can see the lines where I've streaked them across the agar. Once they've grown and divided overnight, I can see spots called colonies. Each colony, which now contains thousands of bacteria, was founded by one bacterium that landed and multiplied at that location on the plate. Next, I take a single colony off the plate and put it into a tube containing a liquid growth media. The bacteria use the nutrients in the media to multiply some more. Over time, I know the bacteria are growing because the liquid goes from clear to opaque. Finally, we're getting to the phage steps. Here, I'm doing a simple experiment to determine how many phages are in a sample. Since I expect it to be a fairly high number, I take the sample I want to test and dilute it numerous times. Then I add tiny droplets of the diluted phage liquid to a plate containing bacteria. The next day, I can count the clear spots produced by phages in each of the places I put a droplet of liquid. Then, using a little bit of math, I can calculate how many phages are in my original sample. Though we can't see viruses, it's still important to study them in the lab. By doing so, we can learn more about the role they play in nature and how they shape the world around us.